Okay, so let's look at question number 1. The diagram shows a light microscope. Which labeled part supports the objective lens? Which labeled part supports the objective lens? So the part which supports the objective lens is part B. The part which supports the objective lens is part B. So part B is representing the revolving nose piece. Revolving nose piece. So the revolving nose piece, that's the part that supports the objective lens. Okay, so let's move on to question number 2. Question number 2 is saying, which of the following describes the similarities between a plant cell and an animal cell? Which of the following describes the similarities between a plant cell and an animal cell? So both have cell membrane and nucleus. Both have cell membrane and nucleus. So the answer for question number 2 is B. Both have cell membrane and nucleus. Okay, let's move on to question number 3. Which of the following statements correctly defines active transport? Which of the following statements correctly defines active transport? So guys, this question is asking for us to identify the correct definition for active transport. So remember what we said. Active transport is the movement of mineral ions from a low concentrated region to a high concentrated region, okay, against the concentration gradient using energy from ATP, which is adenosine triphosphate. Okay, active transport is the movement of mineral ions from a low concentrated region to a high concentrated region against the concentration gradient using energy from adenosine triphosphate, which is ATP. Okay, so in this case, active transport is the movement of molecules across a membrane against a concentration gradient using energy from ATP. Okay, so the correct answer for question number 3 is B. Active transport is the movement of molecules across a membrane against a concentration gradient using energy from ATP, which is adenosine triphosphate. So the answer is B. Take note. Active transport, in other ways you can say active transport is the movement of mineral ions from a low concentrated region to a high concentrated region against a concentration gradient using energy from ATP. Okay, so the answer for question number 3 is B. Okay, let's look at question number 4. The diagram illustrates one mode of enzyme action. The diagram illustrates one model of enzyme action. What model of enzyme action is being demonstrated in the diagram? What model of enzyme action is being demonstrated in the diagram? Okay, so we have got an enzyme, we have a substrate, then inside an enzyme you have got an active site where the substrate is going to fix itself. That's the active site. An active site is a site where the substrate fix itself. Then we have the combination of a substrate and an enzyme forming an enzyme substrate complex. Then we have got the product there. Now the question is saying what model of enzyme action is being demonstrated in the diagram. Okay, So the characteristic of enzyme being demonstrated in the diagram is, you say enzymes work on a specific substrate. Enzyme work on a specific substrate. So the answer is D, substrate specific. Enzymes work on a specific substrate, or you can say substrate specific. Okay, meaning I'll give you an example. Pepsin is an enzyme. So pepsin works on which substrate? Proteins. Okay, so pepsin, the enzyme which is found in the stomach, cannot work on the substrate, okay, which is in the mouth. Okay, because in the mouth you have alkaline condition or alkaline pH. Okay, so the enzyme which works in the mouth is salivary amylase, and it works on which substrate? The substrate is cooked starch. So salivary amylase specifically works on cooked starch. It cannot work on proteins in an acidic condition in the stomach. So we are saying enzyme work on a specific substrate. An enzyme which is working on a protein substrate cannot work on a carbohydrate substrate. One day we are saying enzyme work on a specific substrate. A substrate is a substance on which an enzyme work on. Okay, so each enzyme has got its specific substrate. Okay, that is what we call the lock and key characteristic. Okay, so this diagram, this is what we talked about in grade 10, where we talked about the lock and key characteristic of enzymes. Okay, which defines the substrate specific. So the answer is D, substrate specific.